Let's take today's topic. Last topic we discussed about fertilization, and after the fertilization, as we all know, we have the formation of zygote, and then after zygote is having many changes and it is transforming into various structure, and we have the formation of embryo. So what we are going to discuss, changes from egg to embryo. So after the fertilization, we know that there is a formation of zygote, and then zygote is undergoing into cleavage. We have cleavage, and after the cleavage we have the formation of a structure. That structure we call morula, and then morula is changing into blastula, and then blastula is changing into gastrula, and then gastrula we will have the formation of individual. These are the changes we are going to study. So when we have the formation of the blastula from morula, we have the name that is blastulation. From blastula to gastrula, it means from the blastula we have the formation of the gastrula, and that process is known as gastrulation. And then we have here we have organogenesis. After that one, organogenesis, and this process is responsible for the formation of organs, and after the formation of organs we have the birth of the baby, and then we have the existence of new life. So these are the changes we are happening in case of the humans. So one by one we'll discuss about these changes and how these structures are formed, and what are the consequences. We'll discuss one by one. So when we see where we have fertilization, we have fertilization at fallopian tube. We know that there is a ampulla, isthmus is the structure. So we have fertilization at fallopian tube. Here this is the finger like fimbrial structure. So somewhere here we have that one, we have a sperm, and then we have fertilization. So after the fertilization, what is formed? We have the formation of zygote here. This is zygote, and then this zygote is having cleavage. This zygote is having cleavage, and that cleavage will result into the various kinds of changes, and then we will have the formation of a ball like structure, and that ball like structure we are calling morula, and then after we will have the next step. So we'll focus on this step. We have the formation of zygote, and then we have the starting of the cleavage. It means after the formation of zygote it takes 24 hours, and that zygote enters into the process of the cleavage, and then we have the division of the zygote. So we'll take one by one. So this zygote is having cleavage. So that cleavage will draw here. That structure number A we give this one. So when we saw that one, this is zygote, and zygote is having, we know that after the fusion with the female gamete was having all the structures, that zona pellucida and perivitelline membrane, corona radiata. So all these structures are also intact with this one. So we only the mark perivitelline membrane, zona pellucida, and there is corona radiata. So here we have the division of zygote through animal and vegetal pole. So we will have that division of the zygote through animal vegetal pole. So this is animal pole and this is vegetal pole, and then we have the formation of two blastomeres. What is formed? We have the formation of two blastomeres. So we need to focus on that. Animal pole, already we discussed that pole where we have the formation of the polar bodies, where from we have the entrance of the sperm. So that pole we call animal pole. So first cleavage starts through animal and vegetal pole. So that is the concept, and then after we will have the division. So we'll have to discuss about few points here. We discussed about that plane, animal vegetal plane, and this cleavage is indeterminate. What we call indeterminate. Indeterminate. It means the fate of cells are not decided. So when we do not have any idea about cells what they are going to be, we call indeterminate cleavage. So we have that one indeterminate cleavage, and we have that one division which is known as holoblastic. There is a one word that we write here, holoblastic. So try to get that one. Holo means that whole, means all the content is divided. We have that many kinds of yolk content, many kinds of eggs are there. There is lecithinal eggs, polylecithinal eggs, alecithinal eggs, dependent upon the yolk content. So whenever we have that one alecithinal egg, in case of the ourselves we have alecithinal egg, and when we have that yolk content, it is a very least negligible. So we have the division of full content. So that kind of cleavage we call, that kind of division we call holoblastic cleavage or holoblastic division. So that is the first. What we have in case of A, we can see that one animal vegetal pole, there is a one larger and one smaller, and then after we have a division, and that division will result into first larger will divide, then we will have 3 cell stage. So this is 2 cell stage, and after 2 cell we have the division of larger first, and this division will take through right angle now. So we will have division through right angle. This is 3 cell stage. So right angle division will take place, and simultaneously after the this is B1, after the right angle division we have that larger, we will have division of smaller. So on the whole we will have 4 cell stages, and these 4 cell stages we have the cleavage, 2 times cleavage, and we have a 4 blastomeres. Then we have again division, 8 cell stage will come, and then after we will have 16 cell stage will come, and then after we have the formation of a compact ball like structure, and that ball like structure we call morula. So here we have all these changes. This is the uterine wall, we know that this is uterus, and these are the endometrium. So we know that the function of endometrium, this endometrium will take that structure for the implantation. So whatever changes are happening, these are happening here, A2, then we have B, 3 celled stage. 3 celled stages are there. 3 celled stages are there. Then we will have the 4 cell stage, and then after 4 cell stage, what we have? We will have a formation of ball like structure. Maximum number of that blastomeres can be 16, and then we will have the ball like structure is formed, and this ball like structure we call, these are the blastomere. Still we have filling of blastomere, and then it enters into a stage that is known as compaction. There is a process that is known as compaction, and after the compaction, all the cells are arranging in a proper manner, that is smaller in the margin, and then we have larger, and they are arranging in a proper manner. You can see here, in a proper manner they are arranging. That is compaction we call. So after the compaction, we observe a solid ball like structure, and that solid ball like structure is known as morula. So what we are observing, my dear students, that the size of the zygote is not increasing, only the number of cells are increasing after the cleavage. Here that we have, that we observe, I made this one, that larger one, but the size of the zygote is not increasing. What is increasing? The number of cells inside the zygote is increasing. That is the concept you need to go through. And then we have a formation of a compact ball like structure, and that structure is known as morula. Morula. And that is the first change. So what we studied in this topic, that zygote is undergoing cleavage, and after the cleavage we will have the formation of a ball like structure that is known as morula, and we have all the changes. It means formation of morula, it is happening after the fertilization. It means after the formation of zygote, and all these changes are happening in the fallopian tube, and then this morula will come out, and then it comes into uterus, and then we will have the other changes. It means from the morula we'll have the formation of blastula. The process is blastulation, will take in next video. Thank you everyone.